Hello class, today's topic is topic number 2, which is our unit on the microscope and the cells. The learning targets that we're going to cover in this first video today are listed below. You can see we're going to learn to properly use a microscope to observe a specimen, something that we're looking at, apply cell theory to prokaryotes and eukaryotes, and compare and contrast those prokaryotes and eukaryotes. So to begin with, we'll start talking about the microscope. The microscope that we're going to use is a compound light microscope. That's going to be used to magnify objects approximately 1,500 times at a maximum. So 1,500 times larger than what we see. So we can get images like this. We use slide boxes and things over here. Glass slide to put on there, allow the light to pass through from the bottom, and lenses to magnify the scope. So these are common in schools. That's what we're going to use. Ours look very much similar to this microscope right there. We are going to use magnifications of 40x, 100, and 400x occasionally here. Another type of microscope that has really advanced our understanding of the microbiological world would be an electron microscope. Those use electrons instead of light to magnify structures approximately 500,000 times compared to our maximum of like 1,500 on a light microscope. Scanning electron microscopes scan the surface to learn three-dimensional shapes. So you can see some cool ones here. We have a spider's face up close and personal. We have blood cell over here to see the shape and the structure and kind of see what it really looks like in a three-dimensional world. Just some more pictures here to see a scanning electron microscope and kind of see this is not something that at this point with technology would fit in a school building. Here's a nice beautiful butterfly over here, and then these are all kinds of different pollen and things like that. So it can definitely get cool three-dimensional shapes. Parts of a microscope going to be important when we're talking about how to use a microscope. We're going to use some subject-specific vocabulary, and we're going to go over those things in class for sure. So, focusing a specimen. This is 8 steps on how to be successful using our microscopes in class. So, step 1, always start with the scanning or the shortest objective. The scanning objective is going to be the smallest of these. So, we're talking about our objectives here. It looks like this one that it's actually pointing to is a scanning objective. That is going to be the smallest of them. We start with that one pointing straight down, and then we start to get the image in from there. We're going to use the coarse adjustment knob right away. This is the big one. Coarse adjustment knob is over here. There's our coarse adjustment knob. I'm going to utilize that one and just turn it until we get an object into focus. We're going to move our slide around. We can draw a slide on here. Basically our slide is our glass slide. The biggest danger with a glass slide is if you don't start with the shortest objective like this one and you put the long one on here and then you turn this coarse adjustment knob, you're going to end up pushing this objective through the glass slide and breaking it. So we have a glass slide in there. We're going to move it around until we see something. We're going to use the stage clips to anchor it. Where's our stage clips? Our stage clips are right here. You can see those metal things right there. Stage clips are going to hold the glass slide down. You pick them up, set them on the top. Once you've focused on the scanning power, we can switch to low power. So you'll use this right here, this revolving nose piece. That part turns, and we'll switch the objectives. Then we're going to switch it again to higher power. You continue to get it in focus. At this point, when you have a bigger objective pointing straight down towards your glass slide, you don't want it to punch through your glass slide and crack it. So at that point, the coarse adjustment knob becomes a no, don't touch, and then we want to utilize this fine adjustment knob here. That's a smaller knob, and that's when you have the higher objective. So we turn that one, that'll get into sharp focus. Again, do not use the coarse adjustment knob. We do not want to do that when we have a high objective or a bigger objective into focus. Drawing the specimen, 3 tips. So once you do get into focus, you're going to be asked to draw in a couple of labs we do in class. When you draw a specimen, you're going to use a pencil. Always use a pencil so you can erase shaded areas. pen will be a huge problem for sketching anything. All drawings should include clear and proper labels and be large enough to see in view. So we have our examples over here of our clear labeling, pointing to the actual object. Again, label, label, very clear. They should be written outside of the circle and then lines pointing you to where it is. When you do label it, you should also make sure you're writing what it is you're drawing a picture of and at what magnification. If your specimen takes up the whole viewing field, make sure the drawing does reflect that. So we're going to practice drawing at a couple of different magnifications. We're going to quick discuss here is prokaryotes and eukaryotes and the discovery of cells. I'm not much into history of cells, so we're going to breeze through this one here. What I want you to understand is at some point somebody had to get credit for the discovery of cells, and it was Robert Hooke, a British scientist who first observed and named the cells. He was looking at tree bark, and he looked at that bark and he said, hey, it looks like kind of tiny little rooms or compartments, and referred to them as cells. So this was in 1665 when we had this concept that life was made up of these little compartments, basically. Virchow is the next one. He's a Russian scientist credited for creating cell theory. One of your learning intention or learning targets you're going to have to do is apply cell theory to prokaryotes and eukaryotes and explain how that relates to them. So cell theory is really important because it gives us an idea of how cells actually work and function and gives us some basic rules to follow. The first is that all organisms are composed of one or more cells. That's really important to understand, is that living things are characteristics of life. We talked about it. That's where that comes from. They have to be made up of at least one cell. The next thing is that cells are the basic unit of structure, or the basic unit of organization, structure, and function in living things. And then the final thing is that all cells come from pre-existing cells, so the cells must be multiplying, reproducing in some way. So let's take that information and move along into prokaryotes and see if they do any of those things. So right now this is our types of cells. We have two big categories of cells. We have prokaryotes and eukaryotes. Prokaryotes have no nucleus. We'll start talking about organelles in the next video, and the nucleus is really important for eukaryotes, but prokaryotes don't have one, so it really makes them more simple. DNA is free floating inside the cell. They are the smallest. They are unicellular, which means they're made of one cells, and they're tiny compared to eukaryotes. They have no membrane-bound organelles. Again, when we talk about organelles, it's going to make a little more sense. And they reproduce through binary fission, so they split in half and create two new cells rather than have a more complex procedure. So binary fission is shown down there in that diagram. And the example is bacteria. So here's bacteria presence. Eukaryotes, we like to see them as a little more complex kind of cell. DNA is held inside of a nucleus. It contains membrane-bound organelles, so there's a lot of stuff going on inside that's a little more complex. They're much bigger. It can be 100 times bigger than a prokaryote. So a eukaryotic cell can be bigger than a prokaryote by 100 times. They can be multicellular or unicellular, so they can be made up of many cells, or eukaryotes can be made of many cells or just one cell. And then they reproduce through this process of mitosis, which we see over here. A few more steps involved. Essentially the concept is the same, but because they have more stuff inside, they have to do a little more complex of a process in order to split into two cells. But essentially you start with one and move into two. There's just more stuff in here to keep track of. And you can see membrane-bound organelles in there. All these little things are membrane-bound organelles. And just a quick diagram of prokaryotes and eukaryotes here. So you can see some of the major highlights and structures. Essentially this is, right here, just a sac with DNA inside, and this has a bunch more complex components contained in it.